This lecture covered chapter 10 entitled Sequencing Genes and Genomes. In this chapter we will talk about both the chain termination DNA sequencing, which is also called Sanger sequencing, as well as some different technologies commonly referred to as second generation sequencing or next generation sequencing. We will also cover some of the strategies by which these technologies can be used to sequence an entire genome. Chain termination sequencing, also called Sanger sequencing, was developed by Sanger and colleagues in the mid-1970s. This protocol has some similarities to PCR, but there is only one primer. This means that the copies of the template DNA are single-stranded. Another difference is that a small fraction of the dNTPs are actually D-deoxynucleotides, which means that they have a hydrogen atom here in place of the hydroxyl group in a normal dNTP. When such a nucleotide is incorporated, the product can no longer be elongated because there can be no phosphate bond here. This means that there will be a mixture of different copies of the original molecule, each end by a D-deoxynucleotide. This can be separated by a gel or a capillary gel instrument. And if we label each of these deoxynucleotides differently with different colors, depending on if it is DD-ATP, TTP, CTP or GTP, we can detect as the bands pass the detector which one of these D-deoxynucleotides that end the copy of the DNA. This is a very sensitive separation, so we can detect differences in size of only one nucleotide. The result of this is that we can observe the DNA sequence by the fluorescence of all these different colors here. In the example in the previous slide, the ddNTP was labeled with a fluorescent label. There are also the possibilities to label the dNTPs, in which case you might get a stronger fluorescence, but also perhaps more variable. Or you can simply label the primer that you use for sequencing. Each of these methods have their advantages and disadvantages. This is the most commonly used method actually. Here you would have to use mostly labeled dNTP, which might be more expensive, and in this case you might have to synthesize a completely new primer for each sequencing experiment. Polymerase for sequencing should be carefully selected. In particular, we don't want 3 to 5 prime nuclease activity. We look at the normal activity of DNA polymerase, the synthesis of DNA 5 prime to 3 prime. There are also polymerases that can hydrolyze away the newly synthesized DNA 3 prime to 5 prime. We should not have this in the polymerase used for sequencing because the fluorescent label along with the ddNTP could be hydrolyzed away. Taq polymerase does not have this activity, so we can use that. But in fact, some extra beneficial mutations have been isolated in Taq polymerase. We switch the phenylalanine at position 667 to a tyrosine. We get more efficient incorporation of ddNTPs in general, and we can perhaps use less of this expensive chemical. If we change the arginine at position 660 to something else, we get a more balanced incorporation of ddNTPs. The normal enzyme, or the wild-type enzyme I should say, has a bias for DGTP or DDGTP, which means that the fluorescent curves become very different in strength, and it kind of makes the analysis more difficult. Before starting to sequence a molecule with the Sanger protocol, we actually have to know a small part of the sequence, about 20 nucleotides or so, and we also have to synthesize a primer to bind this area. We usually also try to obtain the reverse sequence by synthesizing a primer for the complementary sequence in order to increase the fidelity of the sequence. We can sequence about 750 nucleotides in one go. This means that if we want to sequence a larger DNA molecule, as this one here, we need to synthesize new primers to sit on other locations in order to obtain the entire sequence. This is a problem if we want to sequence a very large molecule, because we don't know the sequence of this primer before we have sequenced with this primer. This makes the whole process very time consuming. In order to solve the two problems that we posed in the previous slide, the limited range of the sequencing protocol, of any sequencing protocol actually, and the fact that we need to know the sequence right next to the sequence that we want to sequence. Almost all sequencing protocols employ some form of shotgun sequencing. This means that we take a large DNA molecule, we fragment it in a random fashion so that we get all these random fragments, and we locate these fragments in such a way that we have a known sequence just next to the molecule we want to sequence, and then we obtain the DNA sequence of each and all of these fragments, and we analyze them by computer, and we find the overlapping sequences, and we can deduce the sequence of the entire large DNA molecule here. This is an outline of the sequencing of a prokaryotic genome, actually the genome of Haemophilus influenza, by shotgun sequencing. We start by obtaining the genomic DNA of the organism. We sonicate this DNA in order to obtain fragments, random fragments of the DNA. And this DNA, these fragments, we separate on a gel, and we make a size selection here. This size selection is very important for reasons that I will discuss later. We cut these fragments out of and obtain, in this case, fragments between 1,600 and 2,000 base pairs long. And we clone these fragments in order to make a library in E. coli, and we organize this library into these culture plates. And we sequence each of these, in this case around 30,000 of these fragments. We chose this size range because this is precisely the size range where we can cover the entire molecule by Sanger sequencing from each end. We then use primers that are specific for the plasmid vector that we use in order to create the library, and we can get the entire sequence of our insert. We then organize these fragments, the sequence of the fragments in the computer, and we can obtain the entire sequence of the original organism. What was obtained actually in the end of this project was not the entire genome sequence, but 140 contigs of about 12,000 base pairs each. These contigs had no overlapping sequence with each other, so they could not be ordered in the order that they occur in the cell. The reason for this might be that some DNA sequences could not be cloned in the library and they're just missing from the library, or that for other reasons they refuse to be sequenced. It is perhaps so that sequencing more of these clones, or trying to construct more libraries, or libraries in a different organism, could solve this. But at the end of the project, at least each nucleotide had been sequenced 6 times, so there was 6 times coverage, and another strategy was decided upon to close the gaps in the assembly. Ultimately, the gaps in the genome assembly were solved by a combination of a DNA library, a new DNA library made in the lambda vector. As you may remember, the lambda phage accepts much larger DNA inserts than a plasmid in E. coli. Then synthetic oligonucleotides were designed based on the sequence at the ends of each contig. These are 3 of the 140 contigs that I mentioned in the last slide. The library was probed, and in this example here we see one clone that has a positive signal with the oligonucleotide 2. The same clone has a positive signal with oligonucleotide 5, this one. This is an indication that in the lambda library there is a clone that has at least a part of the contig 1 and at least a part of the contig 3, which means that we can order these 2 contigs in this manner. We see here 2 of the main disadvantages or drawbacks of Sanger sequencing in combination with this kind of shotgun approach. One is that there might be sequences that cannot be cloned in any of the vectors chosen, the lambda or any other plasmid, and these sequences cannot be cloned with this, cannot be sequenced with this method. Also, the preparation of several DNA libraries for a sequencing project is very labor intensive and time consuming, and faster methods are actually needed, and we'll talk about them later. The shotgun sequencing strategy, as described, has one major weakness. That is, if our genome has repetitive sequences, as shown here. Sequence 1 is identical to sequence 2, and then there is some DNA in between. If we cut this molecule up in fragments and sequence them, it will be impossible to tell that this part of the DNA was located between sequence 1 and sequence 2, since there will be an overlap between this sequence and this sequence. This means that the strategy as described is really only useful for prokaryotic genomes, because they are relatively free of these repetitive sequences. However, eukaryotic genomes have lots and lots of repetitive DNA, and a different strategy is needed. Since eukaryotic genomes are much larger and much more complex than the typical prokaryotic genome, a kind of hybrid approach has to be employed if we want to sequence this genome by Sanger sequencing. The first step is obtaining the genomic DNA, so this is the same as for prokaryotic genomes. Then we cut it up in random fragments. The difference here is that these fragments are maybe 100 times or more larger than the fragments that were made from the prokaryotic genome. In this case, we make a BAC library. BAC stands for bacterial artificial chromosome, and these are vectors that can take much longer inserts compared to plasmids, for example. After creating a library out of these large clones, this library is organized along the sequence of the chromosome. And when we have done this ordering, each of these clones is treated like as if it were a prokaryotic genome in a way. We cut each clone up into small fragments and we sequence each fragment, and we build up the sequence of each clone from the assembly in the same way as I described. Perhaps a new problem here is, how do we actually do this ordering of the clones? There are several techniques, and I will talk about one that is called chromosome walking. So how will the hierarchical approach help with the problem of repeated sequences? Well, this is simply if the insert size in the back library, in this case, in the library with the large fragments, is large enough to contain 2 of the repeats. This means that there will for sure be another clone in the library that has only 1 of the repeats, for instance. If we look at only this sequence, we may not be able to assemble it correctly because the sequence between the repeats will be lost. But we will have this clone as well that has only 1 of the sequences, and this one will be no problem to obtain the entire sequence. And we have this part of the sequence here, and we can use this information in order to spot the problem with the assembly and correct it. As we might have understood by now, repetitive sequences cause a lot of the trouble in DNA sequencing and sequence assembly. And there are techniques to actually target these repetitive DNAs directly. We can use interspersed repeat element PCR or IRE PCR in order to amplify the sequence between repetitive sequences. If we have a primer for one of these repetitive sequences, usually they are known, we can amplify this PCR product with the same primer. So the same primer sits here and here, and we can spot this or analyze these PCR products on a gel. This makes it possible to make a rough analysis of which repetitive sequences are present on which clones. For instance, these 3 clones here could represent the clones that we saw in the previous slide, and the presence of a band of a similar size in both clones indicate that one of the repetitive sequences is present on each clone at least. Depending on which kind of genome we are sequencing, the genome might contain something called sequence tagged sites, STS. These are simply genes or other sequences in the genome that are known. We can use this information to, for example, design PCR primers for some product that amplifies an STS. And we can use this to make a PCR screening to see which clones that might include this STS. And if we find 2 that does, like in this case, we can infer that these 2 belong together in this manner. As no sequencing method is perfect, the consensus sequence or the true sequence of the genome has to be built up from several reads, ideally covering the same position several times. In this case, we can see that this position here is covered 5 times, 1, 2, 3, 4, 5. And there seems to be a sequencing error here in read 4. It shows T instead of C. But we can deduce that the true nucleotide in this position should be C, because it's simply the majority. We have in this case a depth of 5 in this position. Next generation sequencing is a name that we use for a collection of technologies that have much higher capacity than the chain termination sequencing that we have been discussing. The biggest problem with chain termination is the need for cloning. We need to clone every fragment that we want to sequence. The rest of the protocol has been automated with machines like these ones. You can run several hundred samples 20 times a day using these machines. But even so, the capacity and the capacity is not high enough and the cost is not low enough. So now there are machines like these ones that can maybe sequence 20 million molecules at the same time using a very highly parallel sequencing technology. You can sequence millions and millions of these fragments at the same time at a much lower cost. And the basic reason why this is possible is that the DNA molecules here sit very tightly inside the machine. Here several different next generation sequencing protocols, but they all have a few things in common. The first step is usually that we fragment the DNA using some physical method, and then adapters are linked to each end of the DNA fragments. These adapters are in principle the same kind of adapters that we saw in chapter 4, but the purpose here is different. Then we usually attach these fragments to a solid surface. There are two leading technologies of DNA sequencing, one called Illumina, and here we attach the molecules to a glass surface, and the 454, where the DNA is attached to a magnetic bead. And we use this fixing of DNA in order to pack the DNAs very densely together, but keep them physically separate. The 454 protocol starts with emulsion PCR in order to separate and amplify the DNA molecules that we want to sequence. This molecule here represents all the fragments that we generated in the previous slide. We mix these with the PCR mixture and an oil that can create an emulsion. We mix this vigorously so that we end up with a emulsion of small droplets of PCR mixture in an oil where the majority of the volume is oil. Ideally, we should control the concentration of these components so that each droplet contain one molecule of our DNA library, primers, these green molecules, and one magnetic bead that has single-stranded DNA molecules on the surface. Once we have separated our library fragment here, the primers and the bead, what follows is essentially a PCR, where each droplet in the PCR tube acts as its own PCR reaction. So the first step is denaturing, where we separate the strands of the library molecule. This is followed by annealing, where one of the oligonucleotides on the bead acts as a forward primer, where a new strand is synthesized complementary to one of the library molecule strands. We make a copy of this. What is special with this new copy is that it's covalently attached to this bead here. This will be very important in the subsequent steps. In the subsequent denaturing step, the original strand of the molecule that was not attached to the bead separates and attaches again and can be used as a template for another molecule. And this will go on and on and on until we have a bead that is full of DNA molecules sticking out. What's interesting to note is that they all represent one of the strands of our library molecule. So there are only one part is attached covalently to the bead. Since we have these green primers as well, we also do copies of the complementary strand, so that what we in essence have here is an exponential amplification. Really the exact same situation as if this was PCR. The only difference is that the reverse primer or the forward primer is attached to this bead. If we do this enough times, we end up with this hedgehog structure where the bead is actually full of these molecules. In a subsequent step, we wash away the DNA that is not covalently attached to this magnetic bead. After the PCR, the emulsion containing the beads is simply poured on top of a silicon surface like this one that has these hexagonally shaped holes. These holes are just big enough for one of these beads to enter. You can see a micrograph here on this side. You can't easily fit two of these beads in one of these holes. Then there are also these smaller beads here on the side that you can see. They contain enzymes that are needed for the sequencing reaction. But the important thing here is that each molecule, each library molecule in essence, each bead with a different library molecule is physically separated so that we can analyze them separately. The chemistry of the sequencing reaction, called pyrosequencing, used by the 454 protocol, is quite similar to the chemistry used by Sanger sequencing. The main difference here is that each nucleotide is added one at a time, whereas in Sanger sequencing there are all 4 nucleotides present at the same time. In this case, in this example, we add first a DATP to extend this primer. This one is not incorporated because it doesn't fit, it's not the correct nucleotide, and this will be degraded in a subsequent step. TTP is then added. This one is also not incorporated and it will be degraded. And only when we add DGTP in this case, the DGTP is incorporated into the growing molecule and a flash of light is produced. And then we continue to the next, the CTP, which is also not incorporated, and then we go back to adding a DATP that is in this case incorporated. So since all 4 nucleotides are added in a sequence like this, only one of every 4 will give you a signal. If we look at the chemistry more in detail, whenever a DNTP is incorporated into the growing chain, a diphosphate molecule is secreted or is cleaved off, and this diphosphate molecule is converted into ATP using a couple of enzymes on those smaller beads that we talked about. This ATP is then used together with a luciferase to emit a flash of light, which can be detected by the sensor of the machine. If the DNTP is not incorporated, it will be cleaved off into a monophosphate and DNDPs. which are not substrates for this reaction. So in this case, no flash of light will be emitted. If we look at the technology used by the Illumina sequencing protocol, we can see that it's quite similar. One main difference is that instead of beads, we have a surface. Usually this is a glass surface that has oligonucleotides sticking up from the surface. Another difference is that instead of just one oligonucleotide on the surface of the glass, we have two. And these are very carefully chosen to be complementary to the two adapters that we have on each side of the molecule. We simply pour molecules of our library on top of this surface, and they will stick to random places where they If we instead look at the technology used by Illumina sequencing, we can see that it's in some parts very similar. We have the same kind of fragments with two adapters, one on each end, but we, instead of a magnetic bead, we have a surface. Usually this is a glass surface, and instead of just one oligonucleotide on the surface, we have two different ones. And these are chosen to be complementary to the adapters on each side. So when we perform the same kind of PCR that we saw for the 454 protocol, in this case, the molecule will actually bridge and complement with a oligonucleotide that's sitting quite close to the original molecule. So instead of having a free swimming primer, we have two primers that sit on the surface itself. So this means that all of the molecules will be covalently attached to the glass surface in the end of the amplification. If we do this amplification enough times, there will be copies of these library molecules next to each other, forming little forests on top of the surface. If we start off with a quite low concentration of the library molecules, they will form forests that have some kind of separation between random, but they are separate by statistical reasons. This means that we need equipment that can detect sequencing reactions in random places, and not just inside the wells that we saw for the 454 instrument. The sequencing chemistry used by Illumina is slightly different from the one used by 454. One fundamental problem that has to be solved prior to sequencing is that attached to the surface is not just one strand, but two. So in order to sequence one molecule in an unambiguous way, you have to remove the other strand of the DNA. This is done chemically because one of the oligonucleotides that is attached to the surface is chemically modified so that you can remove it. Here we have all the four nucleotides present at the same time instead of just one by one. They're all linked to a different fluorescent molecule, and we also add a polymerase at the same time. When this is added, the polymerase will take the correct nucleotide and incorporate it, and subsequently you wash the rest of the components away. The only thing you have is the fluorescence of the one correct nucleotide that is attached, and you can measure the color of this fluorescence. Then you remove this stop sign here. This is really a molecule that prevents more nucleotides from incorporating. So by an enzymatic reaction you remove this, and then you repeat the procedure all over again. As you can see here, there's 2 phases here instead of 4, which makes the sequencing slightly faster compared to the 454. Also, you can pack these forests of PCR product a little bit more densely than in the 454, so each instrument has a higher capacity as well. This means that sequencing by Illumina is slightly less expensive than for the 454. Sometimes it's easier to understand something by looking at moving images, and I found these 2 YouTube videos that illustrate in a nice way how the principle of the Illumina and the 454 technology. You should click on these 2 images and go to YouTube and watch them. One fundamental problem of both 454 and Illumina technology is that you add the components for the sequencing one by one, and this essentially makes the sequencing slower than if the DNA polymerase could work at its normal speed. There is a technology called PacBio where you try to solve this problem. Here you use the same kind of cells that maybe you use for the 454 technology, but instead of a sphere here you have one and only one DNA polymerase attached to the bottom of this well, and then you add one DNA molecule, and this polymerase will copy this DNA molecule, and you have attached a glass surface to the bottom or a laser by which you can measure fluorescence in a very, very small volume, only in the vicinity of this DNA polymerase. And each nucleotide is present at the same time, so the polymerase can attach nucleotides as fast as it cares, as it wants. What you do is, whenever a nucleotide is incorporated, the fluorescent molecule is activated, and for a short while it is present in the vicinity of the polymerase, and with the laser detector you can detect a certain fluorescence during a certain time. Then by statistical reasons this fluorescent molecule will swim away from the polymerase, and another nucleotide will be incorporated. What is important to understand here is that we don't really control the speed of this polymerase. We just let it work as fast as it can, and by doing this you can actually increase quite a lot the speed of the instrument. This technology has been commercialized, but it has only a small part of the market due to high cost still. All of the sequencing strategies based on the next generation technologies are necessarily shotgun. This is a shotgun approach. So you start with a long DNA molecule that you fragment, then you sequence each fragment, and you build up the consensus sequence in the computer. There is no way really to make libraries, since you don't make any libraries and you can't use any of the technologies that I already discussed in order to deal with, for example, repetitive DNA. It's rather difficult if the repetitive sequence is larger than the fragment that are used for the sequencing. The problem was solved by using a technology called pair-end reads. So not only do you sequence in the way that I described, small fragments, but you also make a library with much larger fragments, and you can select whatever size you want. And these fragments are so long that you won't really typically get the sequence in the middle, but you will get the sequence in the beginning and in the end. and you know more or less the size of these fragments, so you can use this information in order to resolve problems that come from repetitive DNA. You just need to select maybe several different size ranges of these longer molecules so that you can solve your DNA sequence, even though it might have repetitive DNA sequences. You don't really always want to sequence the entire genome of whatever organism that you add. There are several technologies that you can use to enrich perhaps a type of gene or a family of gene that you are interested in. For instance, if your sample is not a genome but environmental DNA, you might not want to get every sequence of every organism in your metagenomic sample, but only some kind of gene that helps you to distinguish how many organisms are in your sample. And you can use something called DNA capture for this. You design these oligonucleotides that you use to fish out the kind of reads that you want, not reads, but the kind of library molecules that you're interested in. So this is done after the library fragmentation. So you don't really do sequence all the fragments that you had, but only a small fragment maybe. And this means that you can actually, you get less reads, but you can add much more DNA in the initial step, and you can get the information that you want. It could also be other kinds of population, disease genes in population, for instance. Last sequencing technology that I'm going to talk about is radically different from both the 454 and the Illumina that I've been talking about. Here, you don't sequence short fragments, but you sequence very long fragments. And these fragments are the original DNA that you have. They are not copies. So what you do is you split up your original double-stranded DNA molecule with a helicase, and then you push or you pull the DNA strand through a pore, typically a membrane pore protein from some kind of source organism. And you have this whole thing set up in a membrane that is resistant to electrical current. So you can actually measure electrical current through this pore. And this electrical current depends on the DNA that you push through. This is the principle of detection using the Nanopore technology. You have a certain flow of ions if you apply an electrical field across this membrane, and you have a certain current that goes through. If you block the pore, more or less, you get a different signal, you get a different decrease the amount of ions that can flow through. And this depends not only about the size, but also the chemical properties of this molecule that is blocking. And the designers of this system found that this flow depends on what kind of nucleotide that is currently blocking this pore at a certain place. So when you pull the DNA molecule through, you can actually see how these signals differ, and you can deduce what kind of DNA sequence was on the molecule that pulls through. Here's yet another YouTube video. This is very nice and shows very well how this system works. I think it's easier to understand just watching the video. You click on this image and you can go to YouTube and do that. Another attractive aspect of the Nanopore system is that it's very compact. You can actually buy a small machine, a small thing that you can attach to your cell phone or your computer by a USB port. You simply add your DNA here, The preparation of the DNA is very much simpler than for any of the other methods, and it's also less expensive. You can buy this kind of device for about €1,000 or so, and it can sequence up to 30 gigabases of data. So the total cost of the sequencing is quite low. You also had the added advantage that your reads are very long. So the problem of repetitive sequences is much less of a problem. And it's also easier to assemble your reads into a complete genome sequence. Up till now, the problem with this technology has been low fidelity. So you have more errors, maybe 10 or 100 times more errors than for the other technologies. No sequencing technology is perfect, and regardless of what technology you choose, you're not likely to end up with a perfect sequence. You are more likely to end up with a number of contigs that represent your genome. For instance, if we have an organism that has 1,000 or 1 million base pairs here, 1,000 times 1,000 base pairs, we would perhaps end up with a number of contigs where the biggest one is 30 kilobase pairs. We take all these fragments and we order them by size, and we look at what is the size of the contig at 50% of the genome size. This size is the N50 size. So it's the contig, the length of the contig at 50% of the coverage. So you have then smaller contigs on this side and larger on this side. And this is a popular quality measure. It's quite easy to calculate for any sequencing project.